particularly our laboratory last 18 years working on mostly two cancer model. One is breast cancer and another one skin cancer or melanoma. Although we do work related to prostate cancer, cervical cancer and many others, but those are mostly in a collaborative way. So in terms of breast cancer, all of you know that this global breast cancer burden incidence and mortality 2015 to 2024, 19.7 million cases, next decade 10.6 million cases in less developed country like India. Then by 2020 over 1 million cases per year in low and middle income countries. And most common cancer killer among women, breast cancer is the most common cancer killer. 5.8 million women will die in next decade, 3.9 million deaths in less developed country, and more than 1.5 million deaths premature and preventable. So this is the report came by Globocan 2012 IRC. Particularly that according to WHO 2012 reports that here India recorded 70,000 deaths due to breast cancer, China 47,000, US 43,000. And as you can see that among breast cancer there are 4 major subtypes. If I show you the next slide, these are the subtypes. Luminal A, where your estrogen receptor, progesterone receptor positive, HER2 negative. I think all of you, majority of you know that. Luminal B means all 3 positive. And HER2 overexpressing, where ERP are negative, and triple negative, like ERPR and HER2, all 3 negative. These are called basal-like breast cancer. And country like India, we see almost 55% of the patients belong to last 2 category. And particularly triple negative breast cancer, now we can see even Northeast also, they have a high incidence of TNBC, triple negative breast cancer, and those are more aggressive than luminal A, B, HER2 positive. So of course that they are therapeutic targets, they are biomarkers, early diagnostic, prognostic markers and signatures. They are different than luminal A, B and HER2 positive cancer. So now what is important that whoever doing cancer related research, not only breast, any solid cancer, things are little different the way we thought earlier, the way we handle cancer research. So for example, any solid tumor, what you see here, because of inter and intratumoral heterogeneity in the tumor microenvironment, all the tumor cells, what you see, these are not equal and identical. So many of them, they are more aggressive, many of them less aggressive, and most of that active tumor cells which you see in this solid tumor, they resides in the periphery of the tumor. So whatever tumor cells they present in the central mass of the solid tumor, they are not active in nature, they are more passive in nature. So all the leaky vessels, vascularizations, angiogenesis occurs at the periphery of the tumor. Other important point, people thought targeting tumor cells or cancer cells will solve the purpose, but that is not the issue, because now there are many stromal cells. More and more paper coming, particularly outside India, like in case of pancreatic cancer, breast cancer, prostate cancer, lung cancer, etc., that your immune cells playing very important role. That includes macrophage, tumor-associated macrophage, NK cells, T cells, and many other immune cells. That's why today, wherever you go cancer-related, people are talking about immune therapy, because immune therapy, particularly targeting PD-1 and PD-L1, is one of the hot area in cancer research, and these are basically they are targeting based on their antibody-based therapy. Other important point, once tumor develop primary site, but then they metastasize. In case of breast, they goes to the lymph node first, as you can see here, and from there they goes to lung, liver, brain, and bone. Many of the patients die because of metastasis, and country like India, we don't have a good proper awareness and detection system by MRI or mammography, because many of our women, they are not ready to go for the regular screening and testing. So when they detect the cancer, they are in the late stage. They are like a grade 3, stage 3, etc. By that time, the tumor mostly metastasized to other organ, and because of that they die. And so what is important here, the point I like to emphasize, understanding the process of metastasis and that nature of genes and proteins are involved in the development of metastasis entirely different than development of cancer at the primary site. And that area is much less explored, and that is the area many people are focusing. Other important point I forgot to mention here, at the primary site, whatever those mixture of cells, there are some cells they call TIC or CSC, tumor initiating cells, or stem-like cancer cells or cancer stem cell. Again, this is another area is very now exciting and hot. No matter what kind of cancer, people are isolating cancer stem cell and targeting cancer stem cell instead of targeting that entire cancer cells. So that will be the second part of my today's presentation, not in breast but in melanoma, skin cancer. But I have 3-4 PhD students working on breast-derived cancer stem cell, but I will not show those data today. So that type of genes and proteins which we are working last 15-16 years, they are like a chemokine type of protein. They are ECM protein, extracellular matrix protein. One of these protein called osteopontin. By name you can see they express many bone-derived cells like osteoclast, osteoblast, plays a role in bone remodeling. They have some role in cardiovascular system also. But in cancer, they have a multiple isoforms and they are post-translationally modified. So the particular form present in cancer, they are different than what you see in that cardiovascular system or in that bone remodeling. And so this protein you can see biochemically, it has a multiple aspartic residue near the N-terminal, 14 aspartic acid residues. That's why this protein is acidic in nature. It has a unique RGD motif. Many of you know who are working on the drug delivery, you use RGD or cyclic GRGDS as a tag when you conjugate with a nanoparticle, because this sequence recognize integrins. So this is basically integrin mediated delivery, drug delivery, and so particularly alpha V beta 3 integrin. Integrins, you know, they have a 22 alpha subunit and 18 beta subunits. They form heterodimers. They also call addition molecule. So you can see there are many integrins here also. So this protein also interacts with other integrin like alpha 9 beta 1, but these integrin express only immune cells, not in tumor cells, whereas alpha B beta 3 integrin express in tumor cells. So that means this alpha 9 beta 1 integrin interaction with osteopontin, it does not work through RGD motif, but it works through other motif. What you see here, SVVYGLR sequence. And this protein also has a thrombin cleavage site. And other important point, this protein not only interacts with integrin, but it also interacts with CD44, particularly V3, V6 variant. And CD44 also interact with high alloderonic acid, HA. And HA also binds with HAPP. There are groups like Professor Kosturi Dotto and others work on that area. So because of post-translational modification, this protein, once it's post-translationally modified, interact with integrin, but CD44 interaction does not require any post-translational modification. That interaction occurs through C-terminal, whereas that integrin interaction occurs through N-terminal. So there are more than 150 groups all over the world working on this class of protein. There is an international conference every alternative year, etc. So now you can see that what I told you, this protein, no matter whether it interacts with integrin or CD44, it regulates cancer cell migration, cell-cell communication, addition, angiogenesis, production of inflammatory cytokines, differentiations, etc. So while many people working on this field, the paper came from Professor Bob Weinberg's lab from Whitehead Institute of Biomedical Research, MIT Harvard, have shown that systemic endocrine instigation of indolent tumor growth requires osteopontin. What this group have done, they have developed a model, animal model. They injected aggressive cancer cells, one side of mouse, they call instigating primary tumor. So they use in their paper colon cancer, breast cancer, etc. And same mouse, the other side of that mouse, they injected less aggressive cancer cells of the same cancer origin, they call non-instigating secondary tumor. So osteopontin secretes from the instigating primary tumor, move and migrate towards that non-instigating secondary site through bone marrow activation. So that was the novel finding, that's why the paper came in Cell back in 2008. So that suggests that bone marrow derived mesenchymal stromal cells or mesenchymal stem cell, MSC, plays very important role. And now there is a series of paper coming on this area. We also have one paper under review where showing that involvement of MSC in development of breast cancer metastasis, response to osteopontin and some of the chemokine receptors and integrins. But again, I am not going to talk on that. So with that, this last January, I had a meeting with Bob Weinberg, and now he agreed to collaborate with us on this area. He has given us many reagents and cells related to that, and that work is going on. So few years back, we have shown that this osteopontin, tumor-derived osteopontin, in breast cancer, triple negative breast cancer cells, MDMB231, it upregulates VEGF expression. VEGF, you know, this is a pro-angiogenic molecule, this vascular endothelial growth factor. So that we have shown in protein level and RNA level, and we have further validated by overexpressing osteopontin or silencing osteopontin by its siRNA or by blocking osteopontin function using the neutralizing blocking antibody, that yes, osteopontin upregulates VEGF expression. And that ultimately we have shown that this osteopontin induced tumor derived VEGF regulates tumor angiogenesis. This is that in vivo angiogenesis model, and that enhance breast tumor growth in the nude mice model. So then we have sequentially analyzed all these by immunohistochemistry, etc. This paper that time published in Cancer Research, and so I have not shown you all the detailed data because I will be showing some new data. So basically we have dissected entire molecular mechanism, dissected that all that pathways, how osteopontin regulates VEGF expression through this multiple activation of multiple kinases, and followed by transcription factor. There is a crosstalk between these pathways, and through autocrine, paracrine and dextracrine mechanism, this osteopontin regulates VEGF, and it either binds with a VEGF receptor called neuropilin-1 or VEGF other receptor called KDR or VEGF receptor 2, or simultaneously with neuropilin-1 and KDR, one expressed in the tumor cell, another expressed in endothelial cells. So upon binding, it basically regulates tumor endothelial interaction, and that controls angiogenesis, tumor growth and metastasis in breast cancer model. So since we have that angiogenesis model, then we checked few compound, and we have found that curcumin suppress osteopontin regulated VEGF, tumor derived VEGF expression, and that inhibits or blocks tumor angiogenesis that time. So similarly in prostate cancer model, there are other students, they are all in US, I think majority of them, they are assistant professor there. So shown that one of the pro-inflammatory molecules I think your group also, some of you are interested working on this, cyclooxygenase 2. Because there is a specific inhibitor, celecoxib, aspirin, they basically target COX2, COX and LOX pathways and block that prostaglandin E2 production. So in prostate cancer model, Salini has shown that this osteopontin basically regulates COX2 expression through upregulation of kinase called PKC alpha, and this PKC regulates kinase like nuclear factor inducing kinase that regulates IKK alpha beta kinase activation, and that regulates NF kappa B activation, and NF kappa B controls COX2 expression transcriptionally, and that regulates that prostaglandin E2 production. And that PGE2 has a receptor called EP2, EP4. They are GPCR. They are G protein coupled receptors, 7 transmembrane domain receptor. So once it binds with particularly EP2 and EP4, that regulates prostate tumor growth. Again, that another area in cervical cancer model, that other person, he is also now moved Moffitt Cancer Center to Philadelphia. He has also shown that in cervical cancer model, particularly P38 MAPK, one of that important target in cervical cancer, is regulated by OPN, but not by interacting with integrin, but by interacting with CD44, and regulates that activation and expression of serine protease called furin, and that furin basically regulates cervical tumor growth, which he has shown by multiple in vivo models as well as in vitro models, by overexpressing osteopontin or by silencing osteopontin using shRNA. And here that's the time we had that in vivo imaging system where we have used that cervical cancer cells expressing luciferase, LUC containing cervical cancer cells. So we injected to the mice, developed tumor. As you can see the image here, I think pointer is not working. So, and then we tested couple of druggable molecule like a P38 MAPK inhibitor, SB20358, or furin inhibitor that suppress cervical tumor growth. So this has been analyzed because this in vivo imaging, some of you who are working in the cancer or other disease related, is one of that latest technology in terms of screening the drugs in vivo model. So we have quantified this based on this its software and represented here, and also we have subsequently analyzed the tumor volume, tumor size, and from this tumor we have also analyzed that by immunohistochemistry and fluorescence, we check the expression of P38 MAPK, phospho-NF kappa B, P65 furin, their expression much higher in this tumor, whereas that expression gone down in this tumor, where that this is that instrument which we have, and I am a scientist in charge in that facility. It's from Genogen, California. So we had a series of paper on these issues, mostly in cancer research, oncogenic, etc. So now, since we are working on the tumor angiogenesis and in the breast tumor model, so now you know the hypoxia plays very important role, because once the tumor cell they move away from blood vessels, the level of oxygen goes down, and that's why you see that hypoxic environment. And because of that, one of the very important transcription factor called hypoxia-inducible factor, I think many of you know, HIF1 alpha, they start to overexpress, and that controls angiogenesis through transcriptionally regulating VEGF. So one of my other student who now became scientist, Institute of Bioinformatics, Bangalore, she did some work where she has shown that hypoxia regulates osteopontin gene in breast cancer model, and it is known that hypoxia regulates HIF1 alpha. To show that whether osteopontin is involved in hypoxia regulated HIF1 alpha, so she has used that osteopontin knockout and wild type mice, and she has developed mouse embryonic fibroblast MEF cells from knockout and wild type mice exposed under hypoxic condition, and found that wild type derived MEF, once she exposed under hypoxic condition, is more HIF1 alpha. So that suggests that osteopontin is involved in hypoxia regulated HIF1 alpha protein and RNA level. So then she has shown that hypoxia regulates osteopontin gene, and that controls some of the downstream molecule like AKT activation, etc., and that controls angiogenesis in that CAM assay, where you use the fertilized eggs and then develop this. This is one of the good assay if anybody want to screen your small molecule inhibitor or extract which has anti-cancer or anti-angiogenic activity. Is one of the very good assay where you can quantify this. So here she has shown that once she overexpress one of that breast cancer cells like MCF7 and exposed under hypoxic condition at 48 hours, so there is a more blood vessel formation as compared to 0 hour. And these are all negative and positive controls. She has quantified this, but I am not showing those data here. So again, she has shown that by loss of function and gain in function, that MCF cell does not express much osteopontin. She overexpressed and injected to that not-skid mice, developed tumor. So that osteopontin overexpress MEF enhanced tumor as compared to MCF alone. But NDA231, this is aggressive breast cancer cells, that developed tumor itself very high because these are very highly aggressive as compared to MCF7. But if she silence that osteopontin by its shRNA by making a stable clone, that then tumor size gone down. So then analyze that expression of osteopontin, HIF1 alpha, VEGF. In this tumor they are higher, and in this tumor they are lower. So then in collaboration with that our hospitals in Pune, Rubicon Clinic, where these doctors, oncosurgeon, oncopathologist, they are there. They provided us breast cancer specimen with total studies. He has used 53 patient sample, categorized into 4 groups: peripheral tissues, fibroadenoma, ductal carcinoma in situ, and infiltrating ductal carcinoma. By immunohistochemistry, she checked the expression of HIF1 alpha and osteopontin, found that expression of these genes are very high in infiltrating ductal carcinoma as compared to peripheral tissues or fibroadenoma. So that suggests that this hypoxia regulating osteopontin gene through this mechanistically through this pathway, and that controls HIF1 alpha dependent VEGF expression, and that regulates tumor growth and metastasis. So this paper also came in oncogenic. Another student who moved to Southwestern University, Dallas, Texas, she worked on the tumor immunology aspects of osteopontin. She has isolated macrophage from multiple sources, and once interacted this macrophage with the tumor cells, then macrophage become tumor associated macrophage, and that start to overexpress osteopontin. And as I told you, that interact with alpha 9 beta 1 in tumor associated macrophage, and that regulates that melanoma growth and metastasis. And even that some of the patient sample in malignant melanoma, she found that infiltration of this macrophage to the side of that malignant melanoma, which she has proved it by macrophage specific marker, which is CD68, and co-localization of osteopontin with a macrophage specific marker, and as well as there is a more angiogenesis because CD31 is angiogenesis specific marker. So with this series also we had a series of paper in oncogene and others. I'll talk a little bit about nanomedicine cancer, then I'll talk a few slides based on that our cancer stem cell related one. So nanomedicine cancer, I think you have here a department who is handling a lot of material science related activities, but nanomedicine playing very important role, particularly multifunctional therapeutics in cancer. And here you know there are drugs already in the market like Abraxane, which basically tagged albumin with a paclitaxel, and the company who marketed from US, Abraxane, and that generic form of this is manufactured by Biocon Bangalore, and some other company. So this is working actively for the treatment of breast cancer. Particularly if you see that why this signaling pathways is very important, not only cancer, other disease as well, but particularly especially cancer. You see whether molecules like a VEGF receptor or some of the downstream like a RAS, RAF genes, each of that already targeted by their antibody, and many of the drugs today available for the treatment of cancer, these are nothing but monoclonal antibody of RAS or RAF or VEGF receptor, or some of them they are small molecule inhibitor targeting these kinases. So now, based on these available drugs, what people are now moving further, they are selecting some nanoparticle which is more biocompatible, biodegradable with this kind of drugs, and delivering in a particular cancer in a more targeted way. And that's why this field becoming more and more important. With this, we also started a project in collaboration with IIT Delhi and V.S. Chauhan, ex-director ICGV, NCL, and few others. But here, size, toxicity, etc., these are important issues, those who are working in the nanoparticle. The project which we are running our own, where we have selected cytosine type of nanoparticle, activated with a chemical crosslinker like SPDP, and further chemically conjugated with a cyclic RGD peptide. You can see this is the conjugation. And again, that pointer not working. So now this conjugated cytosine particle we tag with a hydroxytamoxifen in ERPR positive breast cancer, or paclitaxel, or some of their small molecule inhibitor, and we have delivered, and that line of work going on. This is the work done in collaboration with V.S. Chauhan, paper came that time, nanomedicine. The girl who worked, now she becomes scientist of NIST Mohali. So what we have shown, this animal model studies done in our laboratory, that when doxorubicin tag with a Dr. Chauhan's peptide called R delta F, which further tag with a folate, that shows that fairly good anti-cancer activity, which is the last mouse what you can see, as compared to free drug alone or free peptide alone, etc. So this has been tested in our laboratory. Now the last part of the talk, I'll take maybe another 5-7 minutes. So in case of skin cancer or malignant melanoma, you know this is a major issue, particularly if you go Australia or Europe or US, because of white skin, they develop malignant melanoma because they do a lot of sunburn and lack of melanin synthesis. And it's very aggressive, and many of them they die because of lung metastasis as well as liver metastasis. So the basic theory of this is called ABCDE. So as you can see that when the tumor develops, this will be more asymmetrical. So that's why they call A. Then border irregularity, then C for color, D for diameter, and E for evolution. So these are stepwise process. As I told you that once this cancer develop in skin, so they metastasize to that lung, and many of them they die because of that. So one of my graduate students, he had just completed this project and he just got his PhD degree. So what he asked, that if malignant melanoma cells exist, any cancer stem cell or stem-like cancer stem cell or tumor initiating cells, then how important they are in the development of malignant melanoma, skin cancer, and metastasis. So he searched literature, he checked what are the signatures or marker available to isolate the cancer stem cell by flow cytometry. So he found that CD133, CD20, CD166, 271, 44, MDR1, these are the markers to isolate and characterize cancer stem cell. So then by fax flow cytometry, he has selected mouse-specific melanoma cells. What you can see left upper corner, B16F10, B16F1, these are mouse-specific. And he also used human-specific melanoma cells, A375, SKML2, SKML28. And for your information, patients who develop malignant melanoma, either they have a mutation of BRAF gene or NRAS gene. So some of these cells what we have selected, they are wild type, and some of them they are mutated in BRAF and NRAS gene. And as I told you that we have used all these markers whatever available in the literature. We purchased all these antibodies starting with CD133, 20, 271, ABCG2, ABCG5, VEGF receptor 1, CD166 and CD44, and studied that what percentage of cells they behave like a cancer stem cell in all 5 different cell lines, some of them mouse specific, some of them human specific. As you can see, that percentage varies from less than 2%, it goes up to even CD44 very high, but otherwise up to 20-30%. Then we have gone through the literature and tried to find that whether if we use any double or triple positive cancer stem cell by selecting more than one marker, and that will be more appropriate to study further. But we found that so far, whatever reported, nobody has used any more than one marker to isolate this cancer stem cell from melanoma. So then we selected CD133 as a marker. So here you can see the red indicates that B16F10, then B16F1, then green A375 human, then SKML2 and 28. So basically when you use that B16F10, it shows about 8% of the total cells they are cancer stem cell. So then he used this CD133 positive cancer stem cell and done that microarrays, and globally asked the question, what are the class of genes they upregulate and what are the class of genes they downregulate by Affymetrix platform. And this is the heat map analysis. You can see 42% of the genes upregulate and 58% of the gene downregulate. And when we have done that biological network analysis, we find that there is a good correlation between the upregulated genes and downregulated genes, and they are very much associated with proliferation, metabolic process, apoptosis, signaling pathways, angiogenesis, cell addition, metastasis, which is shown in the purple color left side. So basically, in melanoma cells, because of heterogeneous population, once we isolate the cancer stem cell, and those of you who are working on cancer stem cell, you know, if they are cancer stem cell or TIC, they should be more tumorigenic in nature, they should have a self-renewal property, more proliferative, metastatic in nature, angiogenic in nature, and they should have a drug resistance. Because that's why that many drugs not working, and then patients, they are having a relapse of that cancer. So he has gone through all these steps one by one. First he checked that what are the stem cell specific transcription factor they express in CD133 positive cancer stem cell. As you can see in the left middle panel, that CD133 positive cells, they express more OCT34, which is a transcription factor, cancer stem cell specific, NANOG, SOX10. This has been proved by immunofluorescence and confocal, panel B. And then next, once he has a CD133 positive cancer stem cell versus CD133 negative or unsorted cells, independently injected to the non-skid mice. As you can see that CD133 positive cancer stem cell develop tumor much faster as compared to unsorted or negative cells. And again, these are that level look cells. So we use that our bioluminescence technology and quantify, and this is the quantification here. And so next point, which is more important, if they are cancer stem cell, they should have a long-term tumorigenic potential. So what we have done, Dheeraj has done, from the parental B16F10 cells, he isolated cancer stem cell positive and negative, implanted to the not-skin mice, develop tumor, then remove tumor, then establish primary culture, then analyze the expression of CD133 again in positive versus negative. What you see primary isograph, after that positive and negative cells. Those cells again re-implanted to second set of not-skin mice, develop tumor again, which you call secondary isograft. Again you dissect, remove the tumor, establish primary culture, and then analyze the expression of stem cell, particularly CD133 expression positive and negative by FACS. This all suggests that, and this is the representative primary and secondary isograft. So in both the cases, CD133 positive cells, they are developing tumor faster than CD133 negative cells. So that suggests that they have a long-term tumorigenic potential. Now, since if these are melanoma-specific cancer stem cell, and you know melanoma, they die because of lung metastasis. So here he has injected CD133 positive cancer stem cell as well as negative cells through tail vein. This is an intravenous injection, and as you can see, this cancer stem cell, CD133 positive cancer stem cell panel A, they are directly going to the lung because these are IV injection. And once you remove that lung and do that ex vivo imaging, you can see that here written lung. So we have that ex vivo imaging of that lung from CD133 positive metastatic lung versus negative. Then we section, and these are the H&E staining. So you can see CD133 positive lung metastatic sample, they have more micro and macro mets as compared to negative one. And further we have proved by using anti-melanoma specific antibody. So these lung mets tissues, they are showing more positivity in anti-melanoma specific antibody, which is panel C, left panel, as compared to the negative one. So now, since CD133 positive cancer stem cell, they are developing tumor, they are having a lung metastasis. So then how it is working? What is the mode of action? What is the mechanism? And what kind of drugs it work? So then we gone through the multiple pathways and looking at the literature that particularly cancer stem cell, one of the important pathways is Notch pathway. So we check the expression of Notch 1, 2, 3, 4, etc. Activated Notch called NICD1. So we found that Notch 1 expression is very high in CD133 positive cells. Some of the data I am not showing because of timing. So then we found that Notch 1 transcriptionally regulating CD133 promoter activity. This is the left panel showing that by reporter promoter assay, by making the construct of different transcript covering the promoter of CD133, and this is the chip assay, where you can see that P1, P2, P3, particularly the P1, this is the site responsible for CD133 transcriptional activation. So this is how the model works. NICD1 transcriptionally regulating CD133. So then whatever the microarray related data we got it, so we had to validate this, particularly that angiogenesis and metastatic related gene like a VGF, MMPs, MMP9, MMP2, etc. So we validated by qPCR, real-time PCR, that is panel A, and we quantified and representing here. Not only that, whether those genes are really expressing high level, if we block their expression by their specific siRNA like Notch1 siRNA or CD133 or CJUN, then the expression of VGF and MMP is going down. That has been validated. And then we have further proved that these both Notch1 and CD133 basically regulating the interaction between endothelial cells and cancer stem cells by multiple ways. We have shown that. So in between, we have now, since we have this model, and now we search for some of that inhibitor which can block Notch pathway. And couple of years back we have shown one of this molecule, androgaphilite, it in Bengal it's called Kalmeg, from this plant Androgaphis paniculata. It suppressed breast cancer growth by inhibiting osteopontin expression through this pathway. So we tested this molecule in our CD133 positive cancer stem cell. We find that andrographilite basically suppress cell viability in CD133 positive cells more, and also it suppress that CD133 positive tumor growth with two different doses, which is panel B. And also andrographilite basically abrogates Notch1 dependent MAPK pathway in CD133 positive cancer stem cell and cancer stem cell mediated tumor growth. This all has been shown here. I'm not going through details. What is important, as we have shown that CD133 positive cells, they metastasize to lung if you inject through tail vein IV. So in that lung mets model, we have given andrographilite with 150 milligram per kg body weight. This inhibits lung metastasis. So that is that important finding of these studies. And the way it works, Notch1 basically activates NICD1 through enzyme called gamma secretase, and they are gamma secretase inhibitor, GS9, GS10, etc., GSI9, GSI10. So we find that andrographilite behaving like a gamma secretase inhibitor, and that is suppressing that cancer stem cell mediated lung metastasis, which has been shown by immunohistochemistry here. So basically, as I have shown you that specific ligand called JAGED1 or delta, it binds with a Notch1 receptor that regulates that activated notch called NICD1, and through this gamma secretase enzyme, and this NICD1 transcriptionally regulates CD133 expression, and this CD133 basically controls MAP kinase activation pathway, which I have not shown you the data because of lack of time. Basically, it regulates MEK36 activation followed by P31 dependent AP1 transcription factor AP1 activation, and that regulates many genes, angiogenesis and metastasis specific genes like MMPs and VEGF, and that controls angiogenesis, metastasis and tumor growth in melanoma model, and that expression of OCT3, 4 and nano goes up, and also this regulates some of that other EMT specific gene, epithelial specific gene goes down like E-carotene, etc., and mesenchymal specific genes, N-carotene, vimentin, they goes up, and some of that other transcription factor like HES1, etc., is goes up. So that also we have shown, but I am not showing this data here because shortage of time. So this paper just recently came Journal of Investigative Dermatology, and because of this druggable molecule, we don't call it drug because it needs a lot of further experiments. And this couple of newspapers, they actually read this and they reported. And what is missing, we have used, I am not showing you the data, there are drugs today available for the treatment of malignant melanoma patients like a dacarbazine, dabrafenib, etc. So we have tested those drugs in our CD133 positive melanoma specific cancer stem cell, and in combination or alone, we have tested along with the andrographilite, and we find that andrographilite working in a similar way the way dacarbazine or dabrafenib work in that melanoma. So now we are moving further. In between that director NCL, who recently joined from Hyderabad Central University, Dr. Ashwini Nagi. So because one of the collaborators of this paper, as you can see, Dr. Tulsiram from NCL, he's the guy earlier purified and chemically characterized that andrographilite. So then this director also, he also worked on that andrographilite. So now we submitted a joint proposal to CSIR, is a FTT proposal, and trying to have this andrographilite and its formulation for the next stage. So that is going on. With that, some of the collaborators and students here, some of them here, but many of them they move to US and some new join. With that, I'll stop it. Thank you very much.